having um, we're having several attendees already, and I'm sure several more will join as we do the introductions. Um, those of you who were on the last session with Jim will remember just how challenging and stimulating it was, and the fact that our discussion went right on, and so much so that we've invited him back to be able to even answer further questions. And the other thing that had happened last time is around 5 of the team were on a plane down to ISSX in Johannesburg, which was a particularly wonderful meeting, and it was really, really encouraging to be there and to have about 6 or 7 Ugandan pharmacometricians really holding their own at an international conference. And to me, it felt like one of those milestones that just shows how far we've come as a team. But what it did mean is some of the others have missed the first time, although the recording has been available. So I'd like to introduce those of you who don't know Jim Bosley. Jim is one of those people where if I was to properly introduce him in the American style, I think we'd probably take the first 15 minutes because his CV is as long as my arm. But he spent more than 20 years advancing systems biology modeling and advocating for its adoption in pharmaceutical research. He's been involved in several companies, which he may well mention as we get going, and he's had many senior leadership roles. And I first met Jim relatively recently last year at the ACOP conference, and we had quite a long chat about emerging pharmacometricians. We talked about the group, but we also talked about this question of QSP. And I had noticed at that time there'd already been quite a little bit of debate about what is QSP and where does PBPK stop and where does QSP start? And is QSP just a more parameterized PBPK model, or is it a fundamentally different discipline? And is there a right or wrong? Is there some scenarios where you would use one and not the other? And we had a really great conversation. In fact, we had several great conversations. And so this webinar is an opportunity to open up that conversation to the group. So I'm going to invite Jim to share his slides, give us a bit of a talk, and then we'll open up to a free time of question and answer. As Gabe has already said in the chat, and to those of you that might have just joined, please feel free to use, I think, the Q&A feature on the webinar. You should be able to see that. The Q&A is slightly better than the chat for asking questions because we can actually answer them quite specifically, whereas if you put things in the chat, they can sort of float up and get a little bit lost. So feel free. Remember, number one rule, there is no such thing as a stupid question. If you've got a question, it deserves to be answered. So Jim, over to you.
Thank you, Katrina. I'm reminded of a talk I went to when I was a young engineer. Henry Kissinger was giving a talk to our company. I worked for a very, very large construction company, and he'd been invited in, and the fellow introducing Henry Kissinger said, blah, 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 and really the man needs no introduction, Henry Kissinger. And whatever you think about him, people applauded, and he got up, and whatever you think about him, he had a sense of humor. He said, it is interesting to note that while it may be that I need no introduction, there are a few people that enjoy being introduced as much as I do. So he had the crowd there. I am going to talk today about, we're going to reprise the earlier talk. I've added some things. I've thought of some things. I have to apologize if I'm a little rough today. I drove about 6 hours yesterday to go to a conference on virtual patient validation and verification held in northern New Jersey. That's about 2.5 hours drive from my house. And it was a very good conference, but it was pretty brutal getting up there and back. But it gave me some ideas. Today's talk, I've gone through and I've reviewed it. I've tried to add a few little things. There's going to be a lot of repeats, but I'll probably say it in a different way, knowing the way I think. So you may get a completely different perspective this time. And if there's questions or comments, please feel free to jump in and ask. And I also have some topics at the end. The conference yesterday was so good that I thought, boy, I could give a talk just about this conference, and so I have some ideas for maybe things we could talk about moving forward if you want. It's up to you. So the question I was asked, make sure I get this right. Yeah, was when do you choose PBPK or when do you choose QSP? I, hearing Katrina's introduction, I was thinking, well, really, QSP is really PBPD, right? It's a physiologically based pharmacodynamics, and then PBPK is what we think of as, ADME, absorption, distribution, metabolism, excretion, but with a more physiological focus. Um, and that really, actually, the question kind of begs, well, when do we do things like NCA and just normal PK and pop PK? And so we basically, in fact, I'm going to stop here. I'm not going to say we did. I'm just going to give this talk as if I'm giving it the first time. That'll make it a little bit easier, I think. So when we're modeling, we model depending on context, and all of the things that we're dealing with typically involve decisions. What decisions are we going to make? What decisions are we going to support? And so these might be things like which pathway should we research for a specific drug? Which target should we look at? Which compound is the best? And so on and so forth. Moving into clinical trial design, what is the best design for our trial? Do we have an all-comers trial? Do I have a segregated trial focused on specific inclusion exclusion criteria? There's a lot of decisions, go or no go, and you see the different phases of drug development: phase one, phase two, phase three, phase four, which is post-marketing. But at every point along the line, you have a test, a trial, or preclinically we do some evaluations and we say, do we go forward with this or not into the clinic? And then once we've gone forward into the clinic, we have a first phase results, and we make a go or no go decision based upon that. Um, and that's a very high value thing. Another high value thing would be, should I buy or divest a drug? Um, there's limitations you have to consider. One thing I added here was, what data do I have access to? And this is something that's coming up time and time again in our commercial engagements. Is clients will ask us to do something, and they'll say, "Hey, we'll give you data," and then we get into the mix, and they say, "Well, we can't give you that data." They're very, very, if they have an approved drug, a commercial firm is very loath to share any data with you, because think of it. What if a very smart pharmacologist in Uganda looks at the models that they've created and looks at the data and comes up and says, you made an error here, and they're earning $2 billion a year on that drug? That's the scenario that they don't want to see. So, but a lot of times though, even when we have uh, clients that say, "Hey, we need to know about X, Y, and Z," and we say, "Okay, fine, share your data on X, Y, and Z," and they won't. They can't. They have internal barriers. So it's a big deal. Um, one other thing I wanted to point out here is, depending on the decision, if you're thinking of making a decision about which trial design to do, what's my optimal trial design, what you really want is not the model where we're taking, hey, here's the trial, here's the patients, here's the physiological model, here's what will happen in that trial. That's the input versus output. What we really want to do is we want to say, hey, I want healthy people. What are the inputs that I need to use? And that's the inverse model down here, and that's a little bit
So,
different.
Jim,
Yeah,
I
yeah.
think your slides are not showing at the moment.
Hmm,
S-
I am sorry.
no, no, I thought you were just giving like an introduction before pulling up the slides, but
Hmm.
just when you referred to an image there, I thought maybe you're.
I am sorry. Yeah, let me stop and share here. Um,
It worked very well in practice. This is looking good. Yeah, there
you see
you
it now?
go. Well done.
Oh,
Brilliant.
okay. Thank you. Sorry to go off on this. At least, so I think that what I just said is probably speaks for itself, but
It
uh,
does.
yeah, all right. So there's a lot of different ways that we model data that we have, and I want to emphasize this. One way of looking at things is I have a specific trial and I have some data and I'm going to analyze it. And we can do all of these things with data like that. Um, NCA is very simple. It's often done for dose linearity. You move on to compartmental pharmacokinetics. You can do individual patients or you can do population. You can move on to PBPK if you have distributional data, and then if you need to look at some of the complex interactions, quantitative systems pharmacology. Um, it depends on your decision, though, right? So, um, and I realized when I was going through this that a lot of this is basically because I've been kind of Jim the modeling evangelist for 25 years, and there's been significant pushback. I think that I'm probably going overboard a little bit in terms of defending modeling. And for that, I guess it's a good data point for you. There is pushback, but I'm trying to basically convince somebody with these slides that they should do modeling or they should do a specific type of modeling. And it was just an insight that I got when I was reviewing this in preparation for the talk. Um, I changed the priority of this thing. Basically, if you're going to do a model, you want to have an idea of what the costs and the benefits are. And the first question you really should ask yourself if you're going to do modeling or somebody wants you to do modeling is, what are we going to do if we don't add modeling analysis? How would you make the decision without a model? What decision would you make? What are the consequences? What is the cost of creating the model? What is the uh, downside if you make a bad decision? What is the upside if you make a good decision? And there's many other things that you would stir into the pot in terms of determining if you model and how you would go about modeling. And a model and the plan really is going to have to include what your goals are, what resources you have, and the timing of the decision as well. So some critical things that must be considered. All right. Again, there are different viewpoints, and the viewpoints interact with people's decisions and willingness to accept specific types of models. And this has an historical basis. I would say that there's a statistics-centric viewpoint. Nothing wrong with it. It's a viewpoint based upon certain assumptions. A lot of those assumptions go back to Carl Pearson in England in the early part of the 20th century, um, where Pearson had, I mean, actually religious kind of philosophical viewpoints that he was skeptical about causality from observational data. Um, I would mention these days, a lot of companies are taking observational data and creating synthetic control arms for trials, and basically they're touting the fact that they can create um, essentially a blinded control arm from observational data. So that's kind of a whole research area, and becoming a big business. Um, Still, Pearson was skeptical about causality, and he developed some ways to make sure that you could validate or verify a causal chain, and the randomized controlled trial was the gold standard. Um, if you have a a well-controlled, adequately powered trial, and you've done statistically rigorous analysis of that trial, you can say a lot of things about the people in that trial and that specific dose and that specific drug. You can be very, very certain of saying certain things. You are dealing with typically with a model with, 5, 10, 20 parameters maybe at the high end and a few compartments, and you might have 20, 30, 50 patients worth of data, or, maybe only five. But the point is that it's a parsimonious model, a small, parsimoniously parameterized model, and you can say a lot about what is happening with the sampled population. If you're extrapolating that to some other population, you're doing something other than statistics. And so the statistical constraints, in my view, don't apply. But that's an argument you'll have. Um, and also, it may be in a case where if you're trying to make these decisions properly, getting sufficient trial data at a specific point is too expensive or will take too long. Um, so, as an engineer, I like a mechanistic standpoint. That's my training. Um, and this is, pardon me for being so wordy, but this is an argument. This is the 30-second elevator pitch that it takes about 2 minutes. So I'm sorry. But the point is that if we have a complex system like physiology, it is impossible to do clinical tests to understand everything within the body that's going on. Um, that is because there is feedback. That is because there is nonlinearity. That is because there are multiple compensatory pathways. That is because there are time-based and disease-based states that the patient is in. And just to give you like a quick example, you can't do longitudinal needle stick biopsies of the liver every hour for 48 hours. That's no patient will tolerate that. Um, so there's some things you can't get when you have everything wrapped up, especially if you have feedback, because the whole point of feedback is if there's changes in the inputs of the system, the outputs remain the same, right? And so if I give a drug to somebody and there's feedback loops that are operating, it negates the outward effect of the drug. Internally, there's maybe a lot of stuff going on, and that's why we do the mechanistic modeling so we can understand that. So we can't understand the complex physiology because of a lot of reasons. Um, uh, the What we do is we basically take a liver biopsy, put the liver cells on in a petri dish or in a cultured bottle, and we put some magic dust on it, and we say what happens. Ah, okay. So now we know what happens to the binding of that specific magic dust to that liver cell. We can say something very definitive about the reduced portion of the science. That is reduction of science. That's classic. But again, we want to know what happens to the system. So now we have to integrate those. And that's why we have to put these disease models together. And that actually can give us an idea of what's really going on. This goes back, take that and validate with data, and you have a very good insight. This goes back to Hodgkins and Huxley in 1952 in England, looking at a giant squid nerve. And they won a Nobel Prize for that. Good stuff. Um, the other thing about once we've integrated this model is we have places for data to go. We know we know where the binding constant of a specific T cell engager and a receptor on the surface of a cell goes in the model. There are literature data for that specific amount of for the specific thing. So we can get a very good idea of what the parameter values are, what the interactions are. Um, and the key thing I'm trying to emphasize here is compared to the statistics viewpoint where you're taking a very clear specific data set from a trial and you're fitting that to a parsimonious model, which is doable statistically. When you have a 500 parameter or 5,000 parameter model, the statisticians get a little nervous about being able to fit all those parameters. The point is we have petabytes of data that are relevant to those parameters, and we can fit the data then. Another thing that has happened, and this was a lightning bolt for me, a book called The Book of Why by Judea Pearl. And he talks about the causal revolution, and he says basically to predict counterfactuals, to predict an experiment that you've not done yet, you must have a causal model. And or put it another way, you cannot curve fit your way, or you cannot curve fit a prediction of the future. And this was something I was trying to figure out how to say for 20 years, and it just hit me like a lightning bolt. The other thing I added this to the slides. He points out in the Book of Why, Dr. Pearl does, that if you have statistical approaches to things and you control for confounding factors without accounting for causality, you can actually destroy the signal that you're looking for. So causality is really critical in thinking about how our models, statistical or QSP, work. Um, and so again, there can be an antipathy towards large models, though, and just recognize what where that comes from, and be prepared to discuss that with people that might be interested in modeling with you or working with you. I work for a company called Nova. Nova's about a 50-person firm in France. We have about 30 biomodelers. We have a software arm. Um, right now, our work focuses on doing professional services projects for companies. They come to us and they say, "We need to know about X. Can you model X?" And we say, "Sure, sure we can." And we do a model. The modeling process is basically we define the problem, we do the literature search, we create a computational model, we fit that model to specific data sets to calibrate the model to get. Uh, we've used a priori data, the Bayesian prior, if you will, that's the literature data. So that goes in the model. We analyze what parameters in the model are most influential on the output, so we know what parameters we have to nail very carefully, and then we use clinical data to adjust those potent parameters, if you will, to get an outcome where each patient has physiological output. In other words, there's not a blood pressure of 1,000 over 500. All the blood pressures in line, all the glucose, everything that we have in the model is within physiological range and is expected as a clinician would expect it. But then it also fits the variability of the data, and that's critical. And that was what the ISOP conference yesterday was about. By the way, if some of you are members of International Society of Pharmacometrics, that conference from yesterday will be online, and I have the name later in the slides. A very, very useful thing for me. So where are we at here? Moving on. So again, what we do in our work is typically we actually, and I kind of disagree with this, but my colleagues have a reasoning for this. I just went through this morning a slide on PBPK that one of my colleagues put together, and um, our default is really to do a PBPK model. It might not have all of the different, it might not have kidney, heart, brain, muscle, but it might have the tumor and other, or the tumor and adipose and other for an adipose um, uh, soluble drug. So the key thing is we're keeping track of where the drug is, um, in addition to how much is in each compartment. Um, One thing I mentioned in the last slide here is that with complex modalities that are now being used, and I'm talking about um, antibody drug conjugates, for example, or T cell engagers, or bifunctional, multifunctional molecules, PROTAC, all of these different things that might bind different places and have very complex PK clearance, for example. Um, this kind of amps up the need to make sure that you have that you're representing the physiology correctly, which would tend to push for a PBPK model. That is to say, a tumor will have a different expression of receptors that the drug binds to, and it will clear at a different rate in the tumor than it does, for example, on a piece of tissue that does not have any of those receptors. The disease models are basically, I love making the disease models and I love making pictures like this um, because it allows me to learn how the disease works and allows me to learn physiology and biology, which is, I think, fascinating. From a chemical engineering standpoint, we didn't get to study any of that in chemical engineering. I like learning. But you see what we have there. I believe that's an ADC model. We'll go into that a little bit. And then once we have the structure of the model, and again, this is a question that is often not asked. People put together a model and then they say, oh, how do we fit the parameters? How do we make sure that the parameters are right? Well, you really have to make sure that the structure is right too. So it's um, uh, the model structure will collide with data sets, and you'll either get really good fits, or if you don't get good fits, then either your fitting approach is poor, or you need to maybe modify the model. Um, and it's awfully hard once you spend all sorts of effort on a model to say, well, maybe I have to change it completely or a significant change. So there's interesting bumps in the road, but the model basically should um, be used, and then it's going to collide with new data sets, and you're either going to learn something new or it's going to match nicely. We we've calibrate the model with data sets. We compare that then to data that was not used to do the calibration, and that's part of our validation. And um, uh, there's a lot now coming out on validation of these things, of these models. This brings us to the point that none of your models will really ever absolutely represent reality. And this is a famous painting by Magritte. A pipe is not a pipe. This is not a pipe. Um, it's a representation of a pipe, and your models will be a representation of physiology. Is it good enough for purpose? That's kind of what you have to be thinking about throughout the process. Um, so when you're doing PBPK modeling, here's some things to consider. Um, do you need to represent the distribution of the therapy to the tumor relevant tissue or to adipose or however you want to slice and dice the physiology? You want to make sure that it's relevant to the context of use. Um, you have to know what the perfusion is and these types of data. That's very good. If you're doing interspecies scaling, I think sometimes it's easier if you have the tissues represented because you the size of the tissue, the size of a specific organ is the relationship, the ratio is known between species. So this gives you an easier way to scale. Um, let's see um, what was I going to say here? I think that rather than going through each of these points, um, we basically should just emphasize that the effort to create these is greater than it's like a multi-compartment, just fitting the data with a compartmental model. It does require data from humans or animals regarding distribution. It does require data from humans or animals regarding the extraction ratio. How much does a tissue extract it? And again, I want to emphasize with the complex treatment modalities that are coming up, where you have different expression levels and different clearance rates depending on how bound the drug is in different tissues. It's becoming more de rigueur, more needed. Um, I mentioned that in my company, the preferred way of doing things is to start with a PBPK model. I would probably go the other direction, but my colleagues are very successful in what they do, and so part of my job as vice president is to say, okay, okay, team, you get to make the decision about what you're doing. You're getting good results. I'm going to shut up. Um, but this is typically uh, what we do in terms of creating a PBPK model. And it goes back to the original papers by Otto Bischoff and Robert Diedrich at the University of Delaware in the early 70s. Um, Now, QSP modeling, which is what I've mostly focused on the past 25 years, is you're representing the causal mechanism of a disease and of a therapy. My friend David DeGraff says that disease is a breakdown of feedback loops in the body, and drugs are our attempt to overcome the fail safes in the body. So, for example, um, for blood pressure, if you have ischemia in your adrenal glands, you set off what's called a renin-angiotensin-aldosterone axis, and you may not have low blood pressure. You may just be ischemic in that sense. But what happens is the renin-angiotensin-aldosterone system is a feedback loop that constricts your blood vessels through the eventual action of aldosterone. Your blood vessels constrict, your blood pressure goes up, and so you may not have low blood pressure, but the adrenal gland is making your blood pressure high because it's not getting enough oxygen. That's the feedback. That's the fail safe, and angiotensin receptor blockers and ACE inhibitors are the way that we use to break that renin-angiotensin aldosterone progression and not have high blood pressure. Um, I'm trying to I'm trying to come through here. We can read through this. The virtual population creation is still somewhat of an art, and that's an ongoing research topic. So if one of you bright students wants to glom onto something that's really, really difficult, VP creation is there. Um, and I think, well, the key thing is these models support both decisions. You can optimize trial. Yep, we can optimize trials, but we can also gain insight. And we use mostly QSP models. Sometimes we use simple disease models, but most of the time we're using QSP disease models and PBPK models. I think we went through the. Let's see where we're at. We're halfway through here. I'm gonna try to finish up in about 10 minutes, and then we can have a discussion. But I think this was interesting to people last time, looking at some of the things that we're working on in terms of different drugs. Maybe what we can do right now is, I'm pointing to uh, bispecific monoclonal antibodies, um, bispecific T cell engagers, bispecific monoclonal antibodies, um, and ADCCs. All of these things, these complex modalities, may require a more physiologically based idea of how the distribution is going, how the drug is getting cleared from the body as well. So that's kind of an interesting thing to me. Um, we're focused a lot on oncology and metabolic, uh, excuse me, and metabolic diseases. But we have everything from um, Alzheimer's to heart failure to all of these things. So a lot of different things can be modeled. Some examples, this is an atopic dermatitis, so that's an inflammation axis, which is a key axis in diseases. Um, and I think I, obviously you want to make sure if you're giving a drug, if it's a systemic drug, you want to know how much of it gets to the epidermis and the various level layers of skin. If you're giving a topical, like an emollient, you want to know how that diffuses in or how that affects the transfer rate of, say, moisture out of the skin, which is important. So this type of thing, not only is the disease mechanism needed to be modeled, but the uh, uh, transfer rates into the tissue and out of the tissue of not just drug, but also things like moisture are important to note. Um, uh, oh, I think I think maybe it's useful to talk about. A lot of times we're trying to, if we're looking at a clinical measurement, you have this is SCORAD, EASI. These are itch scores that are used in evaluating atopic dermatitis, and sometimes it's not easy to map the disease that you can model with the feeling that the patient gets. Some examples are itch is one, pain is another. There's no analytical analysis that shows what level of pain it is. So what you have when you do pain research is you're saying, okay, on a scale of 1 to 10, or 0 to 10, where 0 is no pain and 10 is the worst pain you can possibly imagine, what is your pain level? And then you get huge variability in scores where, one person tells you, oh, it's 9 every time you ask them. And it's like, well, excuse me, if it was 9, you would be like rolled up in a ball on the floor, rocking back and forth. You're not 9. But I think that's there's a difficulty in there. Another modeling issue that we face, there's a joke, goes back to 25 years. The guy that founded a company called Intellos, Sam Holtzman, we were talking about modeling different things, and he said, well, show me how to model a hallucination, and I'll show you how to model schizophrenia. schizophrenia has a huge set of scores in terms of how they evaluate schizophrenia. The joke was this, though. He said, "Show me how to model a hallucination, and I'll show you how to we can model schizophrenia." I was talking to a guy, I think it was at Merck, and I repeated the joke, "Show me how to model a hallucination, and I'll show you, and we can model schizophrenia." And he looks at me, and goes, "That's what I did my PhD on at MIT." So I thought that was kind of amusing. Um, but tying the mechanisms that we know about to actual clinical scores can sometimes be difficult. Asthma, now, let me just, I think I wanted to get to something else. Metabolic. Um, Let me just talk about um, atherosclerotic cardiovascular disease. This is the disease that I've actually done some work in. I actually worked on with Regeneron, a project where we did modeling of triglycerides and hypertriglyceridemics. There's hypercholesterolemics and hypertriglyceridemics, and they're different. But we did a model uh, looking at a receptor, and the interesting thing was we were looking at ANGPTL3, and there's two other similar molecules, ANGPTL4 and ANGPTL8. And we did all this modeling, and the correlation was very strong between ANGPTL3 and triglycerides. Turns out, though, it was the wrong target. The target actually should have been ANGPTL4, but that was quite interesting. In this model, though, we're talking about is if you have a model that represents triglycerides in the bloodstream, then you're going to have a model where you can start to predict what will happen in terms of atherosclerotic plaques, foam cells, and these types of things, where you're having you're going to have cardiovascular events due to those things. Now, the cardiovascular event versus, say, um, LDL cholesterol is until you get it very high, it's a very shallow curve. So, and it's it's interesting to me just how little at lower or moderate cholesterol blood concentrations that affects CV disease. But when you get up a little bit higher, it is strongly predictive. We did something where excuse me, we did do this. It is absolute.
I hope you're okay.
I'm not dying. I'm okay. I just had to get a little sip of coffee there. We did something where we took the level of triglyceride in the bloodstream, predicted plaque formation, and the plaque formation then predicted CV events. And we were very successful. That it's a very difficult modeling tool because there's subtle effects. But this is something I thought that was very interesting. It's still, I think, given the onset of GIP and GLP-1 drugs that are all the rage here in the U.S. at least, they've taken the world by storm. We actually were modeling those 25 years ago. I think maybe we could have done a little bit more investigation back then. Um, right now, I think I'll just say we do go into the literature. We've created, for example, an AI application where the AI application reads a modeling paper and then creates the ODE model for us in our platform. This saves some time for us. And so, um, in addition, we manually imported a lot of models. And the modus there is we'll read a paper, we'll import it, we'll create the ODE model in our platform, and then we challenge ourselves to reproduce the figures in the original article. And so you can see some of the things that we've worked here on. I'm going to zip down to the end, though, I think. Um, I will say one of the key things in these types of models is that there's always somebody that says your model isn't perfect, which is true. Um, they rarely acknowledge the fact that not having the model is probably worse. Having no model whatsoever and try to interpret petabytes of data is harder than if you have a model to interpret the petabytes of data, even if it's not perfect. But one of the things that we wanted to do to show the power, predictive power of these models is we one of my colleagues, Claudio Monteiro, came up with the idea. He said, well, why don't we predict a couple of trials? So we have a very large and complex model of non-small cell lung cancer, and over 600 ODEs and I think over 5,000 parameters. And we took this and we did some simulations based upon two different trials, AstraZeneca's FLAURA2 and uh, Johnson & Johnson's Mariposa trial for non-small cell lung cancer. And in both cases, we were able to very accurately predict before the data were released what the survival rates would be, what the progression rates would be, and so forth. And so those those have been a very, I think, a big um, contribution to the field, showing that these models are indeed highly predictive. As I say, I just went to, um, oh, actually, this was the Kaplan-Meier curve of our model versus the data, and the line there is our model, and the stair step is the data, and the model was created before any of the outcome data from the trial was there. So, kind of proud of that plot. And here's another example. This is the FLAURA2. I think the previous one was the Mariposa trial. So let me get to the end here. Okay. I'm going to go through here. And I think I sent these slides, so you have them. Let me just do this. The things I wanted to talk to you here about are, um, as I said, the virtual patient population validation and verification is an ongoing issue. It's an exciting issue, very interesting. There's a lot of good work being done in it in both industry and in academia. The best document, strangely enough, is from a devices perspective. The American Society of Mechanical Engineers has a medical devices group, and they came up with uh, a document, the ASME verification and validation 40. It's a number of one of the reports. Very well written. Um, and then there is various guidances for QSP modeling. The conference that I went to yesterday was establishing QSP virtual population credibility assessment guidelines, and that was uh, held under the auspices of ISOP. So the slides for that seminar should be available shortly, and I'd keep your eye open for it. I mentioned that the exceptional complexity of recent treatments, such as ADCs, T-cell engagers, bi and multifunctionals, PROTAC, all sorts of interesting things that people are doing. All of those things tend to push us towards mechanistic representation, not only of the disease, but also of the pharmacology. So I think the trend would be towards PBPK. To answer the original question we started out with is when do we do PBPK? Especially if the PK is related to binding and differentially expressed receptors or ligands. Um, I thought I'd close with one thing. I recently had a conversation. Um, Well, let's see. I'm an engineer. I'm a kind of a geek. So I do weird like nighttime reading. So I'm rereading circuits because I have a master's in double E. I was reading a circuits diagram, and there's a theorem called Thevenin's theorem. And Thevenin's theorem is basically if you take any linear system of resistors, voltage sources, current sources. Any, no matter how complex, as long as it's linear, that is to say, as long as the voltage and the current sources don't multiply each other, they just multiply constants and create current and voltage. You always, in every case, you can simplify that model to the simple model where you have a voltage source and in series a resistance, which is kind of interesting. Makes circuit analysis much simpler. There is a strong drive towards creating smaller models. Once we have, I mentioned 600 ODEs, 5,000 parameters. There's two reasons for simplifying, at least two reasons for simplifying those. One is if we can get a smaller model, it's easier to explain. And our internal clients, the person that was saying we need smaller models is in one of our clients, and she has internal clients herself that she has to explain these models to. And so she was saying I would like smaller models for that purpose. There's another reason for doing smaller models, and that is if you can characterize the smaller number of parameters well in a physiological way, you can create virtual patients much easier. At Nova, we typically don't have this problem too badly because we have very, very efficient ways of generating. I can generate 5,000 or 10,000 virtual patients in 2 minutes. Just press the buttons and they come out. Um, and the virtual patients then have to be tested to make sure when you run the model, they have to be physiological. You can't have blood pressures of 1,000 over 500, but those patients then are rejected. And then you end up with a set of virtual patients, a lot of them, that are physiological and where the simulations match the distributional outcome of trials that we know about. But that not everybody has that, and maybe that's not the best way to go. Maybe it is a better way to go with smaller models. I would shout out to one of my former colleagues, woman I worked with when I ran the group modeling group at Rosa, Christina Friedrich. She's left Rosa now and is now working for Merck, but she's done some really cool work on simplifying models to get um, fewer parameters to allow easier virtual patient creation, and also some methods for doing that, like Monte Carlo tempering, and there's some fairly sophisticated things to it. So that's an area. Model reduction would be an interesting research project. With that, I think it's been a little bit disjointed, and I apologize for that, but I hope this has been useful so far. I guess I would say, what questions do you have at this time?
So thanks very much, Jim. I don't think disjointed is the word. I think it's more like listening to the wisdom of somebody that has decades of experience. And there's, I think each of us listening probably drew something different from that. I was quite struck by, for example, the fact that it's really quite difficult to have a pharmacodynamic marker for conditions where the symptoms are more subjective. So things like pain, like you say, some of the mental illness, how
Yes.
you know, anxiety, I could see that you could go on physiological symptoms, but if somebody just says they're feeling anxious, or suicidality, I mean, how do you measure these things? And that's, you can get quite philosophical about that. And I was also struck by the fact that you could use AI to, to troll the literature and see if there was, say, a new biomarker that had come out, because sometimes these disease states are so complex and there's so much new literature coming out. It's very
Mhm.
possible someone somewhere has found a biomarker that predicts some aspect that, but AI could help there. And yeah, it's opened up my mind, but I will stop talking because I see two questions already in the chat. Um, First of all, from Davies, asking, what are some of the challenges experienced when you're creating virtual patient populations? And I don't know if Henry's first question goes with that. He's asking about the rationale of using a digital twin approach to calibrate a virtual population. So if you had access to clinical data within a defined time window, how long would it take to create a perfect digital twin for an individual patient? So maybe you could speak to some of those about those virtual populations.
Great questions. Thank you for those. Yeah, we just talked about that yesterday at this conference, and one of the uh, people, scientists from Genentech, said let's use consistent language. She was saying that, and digital twins comes to us not from biology, but from I think mechanical engineering or, some of the heavy engineering companies. But she said a virtual population is just a population of plausible patients where the total population seems to match the distribution of the real population. A digital twin is a specific match to a specific patient or specific animal, where, not to use a name, but it might be patient identifier 5244387, and we'll just call them the aspects of 387. So you do need the individual patient data for creating that digital twin, and that's something that we often have an issue with. The Flora2 and the Mariposa non-small cell lung cancer examples I gave, we didn't have individual patient data, which makes that even more. I was really impressed with my colleagues. It was very cool. But if you get the individual patient data where you have all of the covariates, all of their demographic data, all of their specific disease history, and you can actually do tests on specific, we're all variable in many ways. If some of the variability that affects the disease can be measured and you can get that, you can get a much better aspect of digital twin. I like there's another aspect of digital twins. When we do our clinical trial simulations, there's two reasons one might do it. One is to see how a real trial would play out given our assumptions. And the other is to look at the difference between, say, two different treatments or two different treatment arms and know what is happening. In a real trial, the difference between treatment arms, the differences are due to one, the drug and disease difference, or the drug differences, two, the individual differences, and three, randomness, like uh, stuff that you can't even attribute to the patient or whatnot. And so the difference between the drug effects is obscured in a real trial to some extent, right? I mean, that's an issue that we use statistics to to unblind that. If you do a simulation where all of the arms in your simulation have all used the same virtual patients, all of that interpatient variability is gone, and now you're looking at just the difference in the drug effects. So it's a little more sensitive test. It's kind of cool. Um, and so the so let's go back to the original. I want to make sure I'm answering the question. So if you have individual data and you want to look at specific, um, uh, say a specific, yeah, boy, there's just, I'm trying to figure out how to explain. In the early days of QSP modeling, we did not fit individual patients. We fit individual patient types. So a moderately obese, a highly insulin resistant diabetic patient, right? The categorical. And then we would, and because the computational power just didn't support generating 5,000 patients. And we would take those different types and we would use prevalence weighting. How much of the population is moderately obese, highly insulin resistant, and we would multiply the effects and say, okay, here's what we're going to see in the trial. And that was good. I mean, it was very useful. About 2008, Immunetrics, company in Pittsburgh, now part of Simulation Sciences, and Rosa, me and a couple of my buddies, Tehan, and who's now, well, Tehan was at Merck, and my colleague Rebecca Bailey and I, and a modeler named Paul Brajnik, we put together a model. I wrote some MATLAB scripts, and we did a clinical trial simulation of individual patients. Rebecca created 600 virtual patients by hand. Yes, Rebecca, yeah, don't underestimate Rebecca. She's awesome. But we did this, and we presented those results at the Page conference in Marseille. And that was really the first, I think, where we were actually modeling large groups of virtual patients in a clinical trial simulation. Um, digital. So let me push back to the questioner. What is your interest in digital twins versus a normal virtual population? Maybe they want to type in and respond to that. I'll move on to another question if that's okay, and then if they want to get back, I want to make sure I answered their question. Let's see. I see one about kickstarting um, pharmacological modeling. Ooh, I'm going to ask Dr. Okea to write to me. One of my friends is visiting from Germany. He's giving a talk in Philadelphia today, and interesting guy, Korean ethnicity. He grew up in Germany, got his medical degree in Germany, did his residency at Harvard, taught at Harvard for 10 years and medical school. He's visiting Philly today, and let me ask him. I'll ask him for you, and we'll see if we can get a better answer because you have an MD, I don't. You're way beyond me. So, um, let's see.
So yeah, and I think you're also doing a good thing by joining these webinars, and the Nigeria chapter also have webinars, and joining them, asking questions, networking with different people. If the MD is medically qualified, there are quite a few of us that have kind of come from a very clinical background. The exciting thing is we have engineers and we have clinicians, and somehow modeling is where we kind of meet in the middle and try and solve difficult problems. I think also just having that persistent curiosity where you just keep asking, yes, but why, and not being satisfied and wanting it better. Um, but that's a
Yep.
very kind offer, Jim, for him to get in direct touch, because I'm sure you've got lots of wisdom. And
Yeah.
maybe perhaps a final, oh,
Yeah.
sorry.
Go ahead.
I
Go
was
ahead,
going to say
please.
just a final one, perhaps from Joshua, because I know that this often happens when questions. We've got a couple of questions here, and maybe they relate to each other. Joshua is asking sort of almost what's the trade-off between complexity and feasibility? So when do you go for a more complex versus a very simple model? And Henry's question is more, if you're actually working with sparse data, how would you approach the QSP model reduction to retain the accuracy and the mechanistic insight, even if the data you're using to validate it is quite sparse? So I suppose they're both questions about the trade-off between complexity, accuracy, etc.
To answer the second question first, and if I forget the first question, remind me, but I think it's all part and parcel. You're right. Um, there, I don't think there's sparse data on any disease really in the there may be a few diseases, but for most diseases that we're looking at to creating drugs for, there's a lot of reductionist data. Remember, I talked about reducing, the liver cell in the culture bottle. Um, if we're used to having a well-controlled, adequately powered trial, and we don't have that, that's not sparse data. That's sparse clinical data. We still have 400 or 500 PDFs that people have looked at in terms of the physiology of the situation. So, um, in some rare diseases, um, well, for example, the hypertriglyceridemia, that's a fairly rare familial disease, and so, they trying to find the number of, if you get to 10 patients, you may have used up most of the people you'll be giving therapy to. So, um, but if you have sparse data about a specific disease and you need to make a decision, you have to do what you can to leverage all of the data. And so I would argue that sparse data about a specific disease would drive me to make a more physiological causal model, a more complex model. Um, if I was doing a statistical model, I'd do the opposite, right? I would, if all I was going to use was that one sparse clinical result, then I'd have to, 3 or 4 parameters and fit it and see what happens. So, um, I tend to dive into the complexity, and maybe that's just a personal foible. To dive into the complexity. That was the sparse data, and again, I want to emphasize there's maybe sparse clinical data, but there may be petabytes of physiological data. So it's the total data set. Um, um, One of the scientists from Sanofi made the point yesterday about that is you have both settled science or even bleeding edge science published in the literature, and then you have the specific data set that you got from your animal or preclinical or small clinical study. Does that address the? I mean, we're getting to the end of the hour here. I don't want to take everybody's time, but I can talk about this for hours. Believe me.
I know. It's almost like next time we have you talk to us, we almost need to get everybody to submit their questions in advance after we've chewed over a lot of this, because there's a lot of complexity, and you've illustrated to us beautifully just what these models can do, and it's incredibly exciting to see how well that could predict the clinical trial. And it's just fantastic. And you've brought in all these emerging aspects. You've given us a historical context of how you got started. You've brought in some concepts that are probably new to some of the listeners, and you've totally opened our eyes. I think we are at the top of the hour, so we will wrap this up. But perhaps, I don't know, Jim, if there's somebody on the line that has like a burning question, and they were to contact, would it be all right if they reached out and contacted you, or
Sure,
how
sure.
would they approach that?
Yeah, I think Dr. Okia reached out in LinkedIn. I'll send him you have my email. I'm okay if you want to give people my email. Happy to do that. Happy to hear from everybody, and it's a blast to hear people with enthusiasm for what I've spent a career on. So it's somewhat flattering, I would say. So thank you for your interesting questions. Lovely being with
And
everyone today.
and I would encourage you to, if you're not on LinkedIn, do follow Jim on LinkedIn, because he posts some really helpful stuff. And I find those of you that are just getting started, by following good people on LinkedIn, you see a lot of resources, and it's a chance to interact and ask all of these questions. So I'd encourage you to do that.
Yeah, yeah. Um,
Right.
Katrina, thanks for the invitation. Thank you all for being part of the discussion here, and I really enjoyed it. And I'm sure we'll have some future talks.
I very much hope so. Thank you very much.
Thanks so much. Have a good day now. Good evening.
Good evening.